Alright, so let us revise what we have learned in the action potential, and then we will move towards the frequency of action potential. Well, at minus 55 millivolts, there is a depolarization occurring, and it occurs until positive 55 millivolts. And after positive 55 millivolts, which is an overshoot potential, the repolarization occurs and the cell goes back to the state of rest, which is minus 90 millivolts. In this process, there is certain exchange of charges and there is a certain graph that is created. But how this graph is studied? Well, these graphs are studied by inserting a cathode ray oscilloscope. The electrodes are placed on one place or another place to study the action potential and to study the graphs of the action potential. On the basis of the placement of these electrodes, we have categorized the frequency of action potentials. And that's how action potentials are measured. Well, there are three frequencies at which action potential is measured. The first one is monophasic action potential. This action potential is measured when the one electrode is placed inside the cell and one electrode is placed outside the cell. This kind of action potential is studied only once. Only one graph is created of action potential and one change is observed inside the cell because it is placed inside the cell and outside the cell, so only one action potential that is generated, we can measure that for monobasic action potential. So what happens in the biphasic action potential? In this, both the recording electrodes are on the surface of muscle. That means they are outside the cell. This helps measuring multiple action potentials. So that's why it is known as biphasic action potential. Now there are several action potentials going on about inside the muscle fiber. So to learn about them, biphasic action potentials are used, and biphasic action potential is also used to measure heart rate in the form of ECG and EEG. But that are the topic for another video, so we will not be talking about that right now. Let us move towards the third type of frequency of action potential, which is compound action potential. It is usually the summation of all the action potentials produced inside a muscle fiber. It is also situated, the electrodes are also situated as same as monophasic action potential, but this time all the action potentials that are going on about inside muscle fiber are measured. So that is why this is known as compound action potential. So what is the need of measuring this action potential? This action potential is important for measuring the frequency and seeing if the action potential is normal on inside the cells or not. Abnormal action potential can lead to several diseases, so that's why the measurement of action potential is important. And that's why we have talked about the frequency of action potential here as well. That was all about the skeletal muscles action potential. How about a little comparison between resting membrane potential and action potential? If we look at the definition, this means the resting membrane potential is when the cells are in non-excited state, which means they're all relaxed and they're all in the state of the rest. Well, if we talk about the action potential, the cells are in the excited state, or there's a certain stimulus that has been received by the cells. What is the charge on resting membrane potential and action potential? Well, as we discussed before, there's always a negative charge inside the cell in the resting membrane. And in the action potential, these things are getting opposite, which means there will be positive charge inside the cell. We have also looked at that in the graph. Now, what are ions doing inside the action potential and resting membrane potential? Well, inside the cell, there will be more potassium ions and less sodium ions. As we said, sodium-potassium pump was pulling all the sodium ions outside. So this means the potassium ions will be more inside the cell and less outside the cell. And in the action potentials, the tables have turned. There will be more sodium inside the cell and less potassium inside the cell. Lastly, what were the channels that were involved in the resting membrane potential and action potential? In resting membrane potential, sodium-potassium pump and potassium leak channels were involved, and in action potential, voltage-gated sodium and potassium channels were involved, as we discussed in the graph. So this was all about the skeletal muscle action potential. We talked about the steps of action potential, then a graph that explained the action potential, and lastly, the frequency of action potential. Hope this helped. Keep watching sqadia.com.
Empower your future with sqadia.com. Your journey to medical mastery starts here. Delve into a world where learning has no boundaries. Engage with our comprehensive online courses, designed to enhance your understanding and refine your skills. You control your education. Learn anytime, anywhere, at your own pace. Transform complex medical concepts into engaging, understandable modules. Experience learning that is as effective as it is enjoyable. Realize your potential, reach new heights, and be ready to take on the challenges of the medical field. This is not just about education. It's about preparing you for a successful career in medicine. sqadia.com. Transforming medical education. Shaping your future.